So there are many things I thought about describing here because my work kind of covers a lot of things that are related to the course. But I thought I would touch on something that might work best for this audience, which is a theme that I think we see a lot in biological vision, and I think should be relevant also for machine vision and for the current era of deep networks. So I'll try to convince you of that, or at least show you some examples that point in that direction. And I would love to hear your thoughts, especially the negative ones. So I have a friend who says after every talk he goes to his closest colleagues and asks them to tell him what was the worst thing about my talk, because that's actually the informative piece of information. Okay, because everybody's always saying, oh, that was a really good talk. Okay, so nobody can tell me it's a good talk.
It was this beginning part where you asked for the worst comment.
Yeah, right, that was the worst part of the talk. Okay, at least I didn't apologize. There will be no worst part. Okay, so let's get started. So when you were looking at that image of the cows, which is actually shot in Switzerland, just on a pass on the way to the Bachalpsee, your eyes are taking in the light off of this projection, and you're figuring out, information is rushing through your brain and swirling around, and somehow out of all of that electrical activity, spiking of neurons, arises vision, perception. It's really, I always have to keep reminding myself of how miraculous that is. When I tell my children that I study vision, or my dad, or my aunt and uncle, they say, what's to study? You open your eyes and there it is. Or they say, gee, I have a camera. Yeah, it's got a lens and it takes pictures, and isn't that vision? And of course, I think most of you in this room know that that's not vision, and we don't really actually know what vision is. It means to represent things, we think, in some format. It means to extract pieces of information in some format, but we often don't know what the format is or should be. In computer vision, there's a huge history, 40, 50 years of anthropomorphizing the problem, of turning it into some sort of symbolic calculation that we think should be what vision should do. But in fact, I think oftentimes our guesses are crude or outright wrong, not what the brain does. So I'm going to talk a little bit about the biology, and I'll say it in a second. But I'm going to talk a lot more about perception and about representation. So, and I'm going to talk, really the theme of my talk is sort of connecting representations to their perceptual consequences. Okay. So, light comes into your eyes, and the course, you haven't heard a lot about neurobiology or the visual system. We did have great lectures on the first day from the master. You may recognize him, or maybe you don't recognize him. He used to have a beard, a large beard, and this is actually what he looks like when he's back at Stanford. It's very formal there, but when he came to Germany, he put on the Birkenstocks and the more casual shirt. So that's EJ, just in case you hadn't recognized him yet. This is a rendition by Paul Glimcher. It was done at the Cold Spring Harbor course. One of my favorite portraits. I have this on the wall in my living room. Okay. And EJ told us a lot about the eye. He told us about the pupil and the lens, and he told us about light landing on the back of the eye, and he actually did tell us about a bunch of circuitry here that does calculations on the incoming light. First of all, transducing the light into electrical activity, and then running it through a set of interneurons until it comes out as spikes on the axons of the ganglion cell, which form the optic nerve. Right. So you have about 1 million fibers coming out of each eye, and everything you see, everything you're taking in, and everything you're going to interpret about the visual world is going to come over those 1 million fibers. And like I said, maybe it sounds almost like we're pretty much done, but of course we're not done. And for people that study the cortex and that think about further visual processing, they tend to view the processing that's done here as a fairly minimal set of processing. And in fact, that's been embodied by the geniuses at the MathWorks who developed MATLAB, and they built in a simulation of this entity directly into the language. So if you're modeling cortex, this is your retina. It's already built in. It's really simple to understand. It obeys superposition. It's linear. And if you add two stimuli together, the response of this thing is the sum of the two stimuli that you started with. So it's very easy to understand. So, EJ, I think your research program ended in 1999 when the MathWorks introduced this. Okay. All right. So what happens after the retina? Well, the information goes back into your brain, goes through pretty long pathway, actually, because it seems like a strange way to route the information. Your primary visual cortex is back here, and along the way, it makes a stop in the lateral geniculate nucleus, which is part of the thalamus. People have been poking cells in the LGN for many, many decades, and the loose description for those of us that don't follow in detail is that they look a lot like the ganglion cells. Maybe they have a few other additional amplifications of properties that you see in ganglion cells, but it's kind of, you see receptive fields, that is responses to light, as EJ described, that are sort of center surround. There are some cells that like light, some cells that like dark. They're usually repressed or suppressed by things that are surrounding their central region of response. And so that's the rough description. When you get to primary visual cortex, as I hope most of you know, Hubel and Wiesel and then many, many, many others showed us that most of the cells there are selective for orientation, spatial orientation, and we often describe them with receptive fields, pictures that look like this. They have some region of response that's elongated, let's say responds to light. Then next to that will be two elongated regions that are responsive to the absence of light, and so we get the same kind of inhibition, but now it's arranged in this sort of oriented pattern. And these cells, of course, are selective to orientation. We often model them in the sort of modeling community using things like Gabor functions, a product of a Gaussian and a sinusoid, or derivatives of a Gaussian, or things like that. Okay, so that's V1. Then what? Are we done? Of course not. The information goes from V1. It splits, at least this is the simplistic sort of cartoon story. The information splits. This sort of splitting description is mostly attributed to Leslie Ungerleider, who sort of laid out this partition. So it splits into a dorsal pathway that seems to be mostly concerned with representing the position or the motion or that sort of action of things. And then a ventral pathway that seems to be more concerned with form and recognition and maybe things like texture or memory. And those are very, very crude descriptions of what's happening in these visual areas. The way that we know a lot about this flow of visual information is, especially in visual sensory processing, we can partition the visual system into regions. And largely that's done not by tracing anatomically the wires, but by noticing that there are cells whose response properties seem to be localized in terms of the entire visual field. They only respond to one little part of the visual field. And each of these areas seems to cover the whole visual field by laying these things out in a 2D array. So you can think of a convolution, for those of you that are coming from the DeepNet sort of world. The convolution idea seems to be at least loosely in place. And these visual areas, which are here, so this is a macaque brain seen laterally and unfortunately flipped left to right compared to the human brain that I showed you before. And in the back, so on the left side now is primary visual cortex, and then there's all these other areas. If you the cortex, as probably many of you know, is actually it's really a big flat thing, a two-dimensional thing, which is all crumpled up like a piece of paper inside of your skull. But if you take it out of your skull and you flatten it out again, iron it a little bit, you get something that looks maybe like that. Out here are the two retinae that have been splattered flat. And here's the LGN, here's V1, and then there are these visual areas that are color-coded here. And each of these visual areas has a representation of the entire visual field with cells who have localized properties and are laid out in some sort of a 2D array. And so that's really the main reason that we know that these areas exist, because each one of them is retinotopically laid out. Okay, and if we make a wiring diagram like this, yes?
So that weren't the original definitions based on Brodmann's areas. So in other words, based on histology before function was even known? They were taking
Yeah, I mean, I think not all of that, but certainly some of it. I mean, it was sort of there's a little bit of a chicken and egg thing, right? So there were definitely anatomical observations, tracing of wires, but at the same time, when measurements started to happen, people started getting much better at delineating where the boundaries are. Oh, we thought this was one area, but it's actually three, that kind of thing, right? And you can't, and functionally, well, I'll say more about this in a moment, but the functional properties of these things are actually pretty complicated. Okay, so if you make a wiring diagram, this is from the early 90s, Van Essen, Anderson, and Felleman laid out a wiring diagram where each of these boxes is a visual area, each of these colored boxes, and each of these boxes has a size that is roughly the amount of cortical tissue that is allocated in that area, and each of these black things is a connection. Think of it as a bus or a pipeline that is the connectivity from one of these areas to another. The connectivity from these areas is always, there's about an equal number of fibers going forward as there are backward. Neurons, the axons are one way. Actually, the axons themselves can go in either direction. If you initiate a spike at the wrong end, it'll go backwards. But normally, the spikes are initiated in the cell body and they go forward. And so these connections that are drawn here are both forward, and there's a sort of roughly equal size set of connections going backward. You can see that the connectivity is not some sort of a simple follow along the chain up the dorsal pathway or up the or down the ventral pathway, but is much more intricate than that. There are connections from V1 to 8 other areas. There are connections from V2 to about a dozen or more other areas, etc. So it branches and sprouts, and there are cross connections as you go. So it's pretty complicated. Despite this complexity, a lot of us in the sort of theory community are looking to simplify. And one of the great simplifications was this fantastic diagram that Ted Adelson and Jim Bergen drew, actually at about the same time, 1991, which is slightly tongue-in-cheek, that sort of says in a sort of wishful way, what we would like is that the information comes out of the retina and it breaks up into these parallel channels, maybe each corresponding to a different area, and each of those areas is kind of representing a different thing, a different attribute. And we would like that because in terms of the way we were thinking about computer vision, we would like to partition things out according to these attributes and maybe solve different tasks using different attributes. So motion, color, form, orientation, binocular disparity, etc. And then that goes into more processing, and that goes into still more processing. That's the tongue-in-cheek part. And then there's some memory up here. But, I mean, this is almost a sort of computer diagram. It's like when we build a computer, we have a memory, and we have a GPU, and we have a CPU, and we have a bus. But the way the brain seems to be architected is really something that's hard for us to get used to. But the memory is distributed throughout. There's memory in the retina. When I say memory, I mean it in a very general form. I mean memory is internal state that lasts for more than the handful of milliseconds during which the initial visual responses are flowing through your brain. So any part of your brain that has state that lasts longer than, say, 100 milliseconds, I'm going to think of that as memory, and that means your entire brain has memory everywhere, all the way back to the beginning of the processing in the retina. There's memory. And those things happen on many timescales. There are adaptations and adjustments to the properties of visual inputs that are happening over all of those timescales. Some of those are very long timescales, some of them are very brief. Okay, so the memory is not some separate module sitting up here above, strapped on like the Monty Python skit when he gets a new brain in the mail and he straps it on top of his head and he says he can think better already. Okay, so it's not like that. It's actually mixed into the entire system. Nobody watches Monty Python. Nobody probably even knows what Monty Python is. I'm not dead yet. Okay, right. So this is a sort of, the thing up front here is it's a description of parallel processing, right? So you start with this input and you partition it into these streams that are running in parallel. And so that's not quite right because we know these things have to talk to each other, and you could see in that previous diagram that there were these cross connections between areas. Now the other extreme is actually where a lot of us have ended up recently. And that says, well, actually, things are just going in some feed-forward serial stream. They just travel their way through, marching through one population to the next. That's kind of like what happens in a deep net. And at each of these populations, well, okay, there are channels in each stage, but the channels are all-to-all connected at each stage, one to the next. And we march our way through in a feed-forward fashion until we get to some desired goal, which we could just say is perception or visual awareness, consciousness, I don't know what. So that's obviously not really right either. It's not parallel and it's not serial. It's both. And I'm not going to attempt to resolve any of this. I'm just trying to lay this out as a front-end thing because it seems to be a little bit missing in our discussions in this course. Okay. So let me give you a couple of reasons or justifications that one can offer for why just thinking about feed-forward processing might be sensible, given what I told you before about those visual areas and the fact that there's feedback between every pair of areas that are connected in a feed-forward fashion. So the first thing, the thing that people often point out is based on results by Simon Thorpe and colleagues, that doing visual recognition takes surprisingly little amount of time for a human or for a monkey. That you can recognize things in very brief presentations after, I don't know what the number is, something like 70 milliseconds. Does that sound about right? Ruth can give me the answer. 70 milliseconds. Okay, 70 milliseconds is about how much time it takes for the information to hop from synapse to synapse through your visual areas, down your ventral stream into area IT, which is where we think probably recognition is happening for most objects and things like animals and humans. So 70 milliseconds is the amount of time it takes for the initial spikes to show up in IT. And there's not really enough time for round trips. There's not enough time to kind of feed things back and then feed them forward again or do that a couple of times. Those things are happening, but they don't operate in those initial spikes. And so the argument is, and those initial spikes appear to, when you measure them in macaques and monkeys, they appear to show a lot of the same selectivity properties that the later spikes show. So the argument is that at least the initial information that shows up is feed-forward and already contains what is necessary to do things like recognition. So that's an argument that you don't need to think about the feedback parts in order to understand the basic computations that are being done by the brain. And you could even argue that a lot of the feedback parts are just there. They're sort of there to provide context and to do adaptation and learning and to adjust things and to mix in a little bit of memory. That's the context. And so the feed-forward processing will get us most of the way. So that would be an argument, for example, that Jim Decarlo would put forward, and he has put forward, that feed-forward processing is going to get us a good chunk of what we want. Another argument for thinking just feed-forward comes really from signal processing, linear systems theory. If I give you a box and I tell you there's a linear filter inside this box, and you get to make measurements on the outside of how do you characterize a linear system, you put signals in, you measure responses. If you measure enough responses, you can actually characterize the whole system because it has to obey superposition. So what you need is to put in enough responses to span the entire input space as a basis. Once you record the responses to all of those, you can completely characterize what's in the box. Okay, I said that very quickly. For those of you that haven't had linear algebra or linear systems theory, please go take a course or read a book because it is really fundamental for everything that we do in science and engineering. And unfortunately, for those of you that are coming from computer science backgrounds, especially in the United States, it's like an optional course. It's sometimes not even mentioned to take things like this. So I highly recommend, if you're going to study vision, go and do that. Okay, enough of that. Where was I? Right, so here's a box, there's a linear filter in it. You can't filter, linear filters can be implemented feed-forward or recursively. In both cases they're linear, in both cases they obey superposition, and from outside you actually can't tell what the implementation is. You can do things to sort of guess, and you can say, well, it's unlikely that the implementation would be feed-forward, but you can't actually tell. You can't actually tell for sure. So, or another way of saying this is, I can make two boxes. One of them is implemented entirely feed-forward, and the other one has a recursive unit in it, and I can make them function exactly the same. And from the outside, you can't tell which one I'm handing you. So the point is that many recursive things can behave or can be mimicked or at least approximated by things that are just feed-forward. So that's a second argument. And there's a third argument, a more subtle and abstract one, that we can get away with just feed-forward descriptions, and that is that it's called the data processing inequality. Information, when you propagate information through a system, you can't create information. When I say information, I mean information about the stimulus. Okay? So if there's a visual stimulus out there, in it comes, you're going to process it. you're not going to add new information about that stimulus. All you can do as you process the information is to lose information. It's a law of entropy. Okay? So as you process, especially with units that are noisy or faulty or insufficient in order to represent the entire quantity of information that's coming in, you're going to lose information. You're not going to gain information. You're not going to add information. You're just going to lose information. So in fact, if we think about the feedback that would be coming into this system, it's not going to be adding information about the visual stimulus. It might be providing context. It might be saying, well, you happen to be looking at something and it's daytime in Freudenstadt, and therefore it's probably, and there's no noise at the moment, so it must be a lunch break because the construction people are not out there banging away and swinging huge shovels around in front of the glass. So so contextual information can come and change your interpretation of the visual information that's coming in, but they're not going to add information about the stimulus. Okay, so therefore, information about the stimulus is propagating feed forward, even if there's feedback. Yeah?
What about memory?
What about it?
Memory is basically.
Of course, yes. That's part of the contextual information would be memory.
But the contextual information is something that you have presumably over your lifetime, right?
Sure.
So are
but
you
it's
just talking about we can represent the whole lifetime of an individual as a feed-forward thing?
Oh, sure. If you want to really take it to its extreme, right. Sure. But that's not even what I was referring to. I was just saying if you want to talk about the information about the current stimulus that's coming into your eyes, think of it from a Bayesian perspective. There's a prior. That's the memory. That's what you expect to see or what you might see, and then there's the likelihood, which is what's...
the memory on top.
Exactly.
I see. All right.
Yeah.
So we often are very vision-focused, vision meeting.
It's a vision meeting.
It's a large part of our brain, but we have all this other sensory input all the time as well,
Yeah.
which is actually new information, which may be combined with the visual
And is, I should have mentioned that.
like posture.
Yeah, absolutely. So that's an excellent point. So not shown in that diagram from Van Essen, Felleman and Van Essen, was the fact that, of course, you have input from all your other sensory modalities, and at some point that all starts to mix in, and these areas become not just visual, but visual and auditory, or visual and tactile, and especially in animals like rodents, mice. Area V1, we think of it as very visual. For a monkey it is. In a rodent, it's responsive to all kinds of things that have to do with whisker twitching and running and whatever other things the animal is involved in. So in fact, mixing in other modalities is extremely important. But we're focused on vision here, and the things I'm going to talk about are pretty early vision. So I think those really are purely visual for the most part. Okay. All right, so maybe we can get away with feed-forward descriptions, at least in early vision and at least on short time scales, and at least if we're only thinking about representing the visual information. Okay, so the questions we can ask, and actually what I'm going to talk about in my time here is how do populations of neurons encode visual information? I'm not actually going to say that much about that. I'm going to leave that kind of short. How do the representations enable, shape, or limit perception? And how do we use those ideas to improve engineered systems? And I'm not really going to be talking about computer vision. I'm sorry to disappoint many of you in the audience who really want to do computer vision. You want to think about visual cognition and things. I'm going to talk about pretty low-level things like removing noise or compressing images or making them look better, rendering them on screens. So these are more image processing than computer vision. Okay. All right. So what goes inside of those visual areas, and that's what I'm going to focus on. We have this notion that goes back, usually gets attributed to Fukushima. On the biological side, it gets attributed more to Rodney Douglas, who introduced this idea of canonical cortical processing. The idea goes something like this. If you look anatomically at chunks of cortex from a monkey taken from different places, so you can take them from visual cortex or auditory cortex or motor cortex or some sort of decision area like parietal cortex. If you look at a little chunk of tissue, it kind of looks the same anatomically. It's a layered structure. It has interneurons and input layers and output layers, and the local wiring, the connectivity, looks approximately the same. You see the same kinds of cells. You see roughly the same kind of connectivity and layering, and you see that throughout the entire cortical sheet. So the argument or the thought is, well, maybe this is somehow badly placed. Okay. The argument, so the idea is that maybe that little chunk of tissue has some computational function that it performs. And it takes inputs, it processes them, and it sends them on. But it also does some recurrent processing and local adjustments and adaptations and sort of homeostatic regulation to keep things in the right operating range. It does things like that. But maybe it's the same operation performed everywhere over and over and over again. And maybe that's the secret that evolution figured out, how to build a more complicated computational capability. Instead of describing the wiring of every single cell the way it's done in, say, an insect, genetically, what you do is you genetically describe this little circuit, and then you arrange to replicate it. So you make tons and tons of these little circuits, you wire them up connected to their neighbors and also connected correctly internally, and then you let the thing learn from experience. You let some information flow through, and it figures out how to assemble and adjust itself to get itself just right, so it's talking to its neighbors correctly and sending its outputs correctly, and that's it. So that idea that there's a canonical computation, a little unit that gets replicated, is an old idea in neuroscience, and probably it goes back earlier than Fukushima, but like I said, he's the one that usually gets credited for that. So a lot of people have built on that idea, and of course convolutional neural networks are building on that idea. They're saying, gee, I want to do the same processing everywhere. I'm going to use the simplest of all canonical computations, which is a convolution. Convolution says I'm going to take a weighted combination of my inputs locally, and I'll do the same weighting everywhere. I'll do weight sharing so that everybody gets the same weights. That's just canonical computation in its simplest form. Okay. And I think, in some sense, this theory it kind of has to be right. If you just say that the rule is just that it has to be canonical, well, if the canonical thing can be as complicated as you want, and if you don't restrict exactly how local it can be, then obviously I can make it as big and complicated as I want. Of course, I can explain the brain that way. I'll just make each of these things, connect to everything and be capable of computing anything, and of course, I'm done. The real issue is how simple can you make it? Can you boil it down to a little tiny element that does almost nothing, and yet when you throw all these together and wire them up, it does something amazing? And I think that to me is the deep net revolution. We're finally realizing that even with something incredibly stupid, a linear filter and a rectifier, you can actually get an amazing amount of power out of that. Okay. But the brain, I think, gets even more power out of what it does, and it doesn't do something quite that simple. So that's what I'm going to describe. All right. So, that was my punchline. I said it and I didn't pop it up. Okay. So the question is, how simple can you make these elements? Okay. So here's some examples of canonical functional models. This is borrowed from a slide of EJ's that I've modified a bit. So there's a linear, you can imagine a linear thing, like I said, a convolution, for example. So this thing is going to just take a weighted sum, and you're going to apply that everywhere. That's a convolution. An LN model, which really goes back to McCulloch and Pitts, who first described in the 1940s this idea that you could describe neurons as, you could describe their dendrites as doing a weighted sum of inputs, and then you could describe something that rectifies those. In their case, it was just a threshold, and then it puts it, in their case, they were imagining a binary output. But if you wanted to do a rectifier with something that's sigmoidal, the idea would be that the output is a firing rate, something that drives the generation of spikes, and then the spikes are maybe random, but with that rate. Okay. And this is also, of course, I'll say more about it in a moment. These days, we see this motif showing up a lot. It starts out in the retina. EJ already described this, what we call an LN model, or a subunit model. The idea is that you have little tiny things that are taking weighted sums. Those get rectified, and then you pool those together into a larger group, and those get rectified. That is a theme that shows up actually surprisingly often, sometimes implemented with different circuitry, but it seems to have similar functional properties. And you see this showing up in the retina. You see it in V1. It appears to show up in MT, or V5, which is an area that's motion selective. And it looks to us at the moment like it's probably going to be in V2 as well. So that idea, subunit idea, seems to be a common theme, maybe a canonical computation. And there's one more, which is actually the theme of my talk, which is this idea of doing something maybe with subunits, but also with gain control. So what is gain control? It just means that at the very end of all of this, what I want to do is divide or normalize the responses by the pooled responses of a bunch of other cells. And I also included in here a little thing that's supposed to be a feedback connection, because it turns out that in a lot of cases, modulatory influences, these could be coming from memory or reward or demands of a task that I'm in, they could be attentional. A lot of those things can also be described as divisive or multiplicative influences on the response of a cell. So these things are going to come in and influence, they're going to regulate the gain, the responsiveness of the cell, boost it so that it's more sensitive, or push it down so it's less sensitive. And I'm just and those things are coming from a lot of different places, but the simple models I'll talk about, they're just going to come from neighboring cells, maybe cells with similar response characteristics. Okay. This idea actually shows up a lot in the literature. You can see it in early retina literature. There's fantastic models in the 70s and 80s from Jonathan Victor describing gain control circuits in the retina and describing their effect. David Heeger, I think, is the person that really pulled a lot of this together and noticed that, at least for primary visual cortex, you could explain a lot of the nonlinear effects that people had observed in different data sets. You could pull them all together and explain all of them in one shot by just imagining that each cell was normalized by its neighbors. And that accounted for really a pretty remarkable set of, a diverse set of data, and became really, I think, the first place where there was a realization that, wow, this very simple nonlinear thing, if you just add that into the mix, is going to let you explain a lot more. And the same kind of thing, if you now take that simplified normalization model, the style that David and then later Matteo Carandini proposed, and you pull it back to the retina, as I'll show you in a moment, you can actually explain a lot of data. And if you pull it forward, you can explain a lot of data. So,
Why did it happen after the nonlinearity and not before?
oh, yeah, so there are many variations on this theme. I'm showing you one particular one. Depending on exactly how you set up your nonlinearities, sometimes it's better to do it before. In fact, if you do it with an exponential and you do it before, then you can actually make it additive and then run it through an exponential. Now it's multiplicative, although the signals you're multiplying are not the same signals you started with, so you have to think through that. So there are many, many variations, but the basic theme is that these things are there to regulate gain. And whether they come at the very end, or sometimes you can actually build some of them back in here in the subunits. You can imagine the subunits themselves are doing some sort of gain control. And then you have another gain control out here. Another thing I should have mentioned is that the gain control can be also internal and over time. That's basically adaptation. So if a cell fires a lot of spikes, and after firing a lot of spikes, it reduces its responsivity. That's sort of an internal self-gain control, which you could think of as like a point nonlinearity or something where you're just dividing by recent temporal history. That happens. Almost all cells in the nervous system exhibit forms of adaptation, and often over timescales that are, sort of, let's say, 100 milliseconds or much longer, minutes, even hours. So these kinds of things also can be described, at least crudely, as gain adjustments. And so, if you want a sort of one box fits all description, it might be that you can just shove all these things in there and at least approximate them as different forms of gain regulation that are coming at different time scales and different sort of spatial scales. EJ.
Just a quick clarifying question. You spoke about feedback here, but earlier you said maybe we can ignore feedback. Are we talking about here only feedback within the layer?
Yeah, no, that's a good question. So, and this is an interesting place where implementation using feedback starts to feel like you really want to include that. So you can describe normalization properties in a feed-forward fashion, and in fact the original Heeger papers do that. They just take all the responses of all the other cells basically simultaneously, combine them, and stick them into the denominator of something that's dividing the cell's response. David himself and others recognized later that if you actually build a recurrent circuit, you can actually set it up, sometimes not even making it do division, set it up so that it's just doing subtractive things in a recurrent loop, and you can arrange that the steady state behavior of that recurrent loop is actually a divisive form. That is, it looks like normalization in steady state, even though dynamically it's doing something different. So there are many, many variations on this theme, and this is a completely, we're not done figuring out exactly how these things get implemented, and probably they're implemented in different ways in different places. So for example, this whole idea of the canonical circuitry, it might hold for cortex, but when you go back to the retina, the structure is actually pretty different. The cell types and the connectivity and the wiring that EJ described for you is quite different than what you see in a cortical column. So to me, the surprising thing is that the response properties in terms of gain control and subunits actually look very similar. So it's something interesting there, that it might be that there are different ways to implement it biologically, and in different species and different organisms and even in different places in our brains, you might actually have gain control mechanisms that are differently implemented but are achieving basically the same functional properties. So that's a theme that has resonated a lot ever since Eve Marder's work. So Eve shows that, I better not go down this path. It's an amazing story, actually. For the ultra-reductionists in the science, in the biological field, the people that believe that we're not going to understand anything until we get down to the molecules and we understand what individual molecules and channels are doing, Eve tells this amazing story about how crabs that live that have the same functional capabilities in their digestive tract have completely different implementations of those functional capabilities. They use different channels and different ions, and the circuitry is identical. The wiring is exactly the same. Every crab is identical, but they use very different ions and channels depending on, first of all, what their environment contains, but also just seems to be kind of random. They have basically a big toolbox. Maybe it's over-parameterized. So there's not a unique solution to the problem of how to achieve that function. So it's, if your goal is to go all the way down to the molecules, you might actually be chasing the wrong thing because it's not unique. It flops around all over the place. And here again, so the analogy is that the retina implements gain control in subunits, so does the cortex. They probably do it differently, but maybe the functional purpose is similar or maybe very much the same. Okay, so we have an evolution. It's important to remember. So you might think I'm standing here saying, oh, here's the evolution. Look, we're done. We reached humans. That must be this over here. But actually, important, somebody, Felix quoted George Box, right? This is the short version of Felix's quote. Essentially, all models are wrong, but some are useful. So we're going to try to keep them simple, and we're going to try to make them useful. And but they're going to be wrong. Everything I'm going to tell you is wrong, okay? And everything everyone else told you is wrong too. They just didn't admit it. So I'm up here confessing. I grew up Catholic, so it comes naturally. Okay. All right. So the LN model. Well, there's not much to say because most of you know about this, right? It's the simplest neural model. You think of the linear filter as doing what the dendrites do, basically adding up inputs, superposition. You think of this nonlinearity as something that transforms that sum of inputs into a rectified value that is a firing rate, and that's what the cell does. This is the classic model for artificial neural networks. I already mentioned that. If this is a threshold nonlinearity, we can actually think of this as a linear classifier. Right? In a high-dimensional space, let's do a two-dimensional space. Let's say I have two inputs. This thing is basically defining a direction in the space. That's what the linear filter is going to do. And then it's taking a weighted sum, so it's collapsing the entire space onto that direction. It's a projection. And then it's thresholding, so it's making a decision about which side of a boundary you're on. So this is the simplest decision maker, if you like. And also, as some of you are probably aware, we have these nice results starting in 1989 that if you wire a whole bunch of these things up together, you can actually approximate any function. So if you put enough of these together with any reasonable point nonlinearity, you can actually approximate any function to any order degree that you want. Okay? So that's nice. It feels like there's a lot of power in these computations. Okay. So we get to deep neural networks. This is, the topic of the meeting, basically. And I'm going to show a whole bunch of deep net results where deep nets actually don't really work very well. So, and there are things for us to think about. They have astounding capabilities in engineering, but they're often very brittle. People often sweep that under the rug, or they point and they say, oh, I don't really care about this. I think those examples are incredibly interesting and very important. For neurobiology, it's been argued that if you train a deep net, it kind of takes on properties that we see in visual cortex. I think that's somewhat true, basic selectivity properties, but I think we don't know enough about what happens in the later areas to really say that it's mimicking what we see in areas of, let's say, the ventral stream or the dorsal stream. And Nico Kriegeskorta and his group have pointed out interesting similarities with MRI responses and the ability to distinguish between stimuli. I'll show you some results that I think argue that although it goes part of the way, there's a long way to go. We still have a lot to figure out there. And what's missing is a lot of the kinds of things I was just talking about, even in that simple gain control subunit model. A lot of those things that are properties of those models of regulating gain have to do with learning and adaptation and homeostasis, and those things are all missing from the neural network literature. Some of you are probably thinking of batch normalization. Oh, isn't that normalization? Batch normalization is done globally, it's done on batches of input, and then it's frozen once you've finished the training. And the operation of the network does not include any kind of dynamic normalization. At least that's true for almost all the deep neural networks in existence. The thing I'm talking about, just to make it clear, is something that operates all the time. It's part of the network, and it's local. So you're going to normalize or divide by responses of your neighbors, maybe over time and maybe over other channels. And that operates all the time. It's part of the network. It's not something you do during training or you do just to get the thing into the right initial state. Okay. So I'm going to, so you can, is the glass half full or is it half empty? It's both. It's neither. It depends on how pessimistic or irritated you want to be. I'm generally not an irritable person, at least not in public. So I'm going to say that I'm really excited about the deep net revolution, even though I find it a little bit frightening. What I'm going to talk about is going to focus on gain control and perception. Okay. So let me say just a few words about this gain control thing and where it comes from. Since you haven't seen much physiology, I probably should not spend too much time on this, but yeah, okay. I'll just, I'm going to skip past the physiology I thought I would sneak in, and I'll just say this. So I already mentioned that gain control is normalizing. Here it's shown before the nonlinearity. You were asking about that. This is a ubiquitous property of sensory neurons. Basically everywhere we look, in the retina, in the LGN, in V1, in area V5 or MT. We see this also in area V2, and I think we're going to see it in other parts of the visual cortex. It's also seen in other sensory modalities. So you see gain control things going on in the auditory system. You see them in the olfactory system. You see them in other species. You see these things in flies. You see them in cats, in monkeys, and mice. So I think, for me, it's an essential property of neurons. And one argument for that is that they have to handle the fact that they're going to get inputs that cover a pretty big range, and they have to keep themselves operating in a reasonable range, so they have to kind of dynamically adjust their operating range. Think of your, this is an old idea in engineering, so think of your video camera or something that's recording sound. If I'm in a video camera in this room and I'm over here taking pictures of Alyosha and I swing it around toward those windows, there's incredibly bright light coming into those windows. It's orders of magnitude brighter than what's over here in the shadows where Alyosha likes to lurk. And so what does the camera do when I swing it over here? Well, it's basically averaging, it's making a measurement, a calculation of the average amount of light that's coming in and hitting the sensor, and it says, whoa, too much light, and it backs down the sensitivity, right? And you see that when you're watching the video, because initially it's completely blown out, and then it comes back down, and you can see things again. Your eyes do that too. You know that if you go into a dark room, you can't see for a minute or two as your pupils dilate and your retina and your entire visual system kind of adapt to the dim light conditions. So you are doing this kind of, we've been doing this kind of gain control in engineering for like 50 years. It's an old idea. In your video camera, it's done globally, and it's done for the sort of mean light level, and it's done over time. The thing I'm talking about is something that will be done locally, not just over time, but also over space. Okay. So dynamic range adaptation, intensity invariance. So if you do normalization and you divide out the intensity of the signal, what you're doing is you think of it as taking vectors of response and making them into unit vectors. What we mean when we're talking about linear algebra, when we say normalize a vector, you make it into a unit vector. So now all the vectors live on a sphere, and the calculations you're going to do have to do with angles on the sphere rather than positions in some giant space. And that changes the way that you think about the computations and the kinds of things that you're able to compute and that you want to compute. It also means that you've lost some information about intensity. So you've thrown it away. That's what I mean by intensity invariance. So you can think about whether that's a good thing or a bad thing for a visual system. And my first student, when I moved to NYU, Odelia Schwartz, and I did a project showing that when you do this kind of thing, if you do the weighting correctly with the neighbors, you can actually greatly reduce the statistical dependencies in the responses of cells, for example, in primary visual cortex. And that is very much a property of natural scenes, as Bill was talking about. So there's superpositions of orientation and things that happen because of the structures in natural scenes, contours and edges and lines. And those things lead to dependencies in response of linear filters that might be measuring those things. And you can eliminate a lot of those dependencies by doing this kind of divisive thing. This is not just decorrelation the way we'd normally do it when we're thinking about second-order statistics and Gaussian statistics. This is sort of higher-order statistics that can be removed by doing this kind of normalization. Okay. So let me, since I'm obviously going to run out of time, EJ's back there snickering because he knows that that's a line that I put out on every talk I've ever given. Fine. Let me jump forward. So there's a gain control model for a retinal ganglion cell from 1987. This is Jonathan Victor, who put forward this idea that, so you measure things over space, separately you measure them over time. This is a biphasic response over time. And then you rectify this and feed it back through some sort of a time delay and smoothing circuit. And then you reset the gain according to that little feedback loop. Okay, so that's a version for the retina. Here's one for the LGN, and I'll come back to this model later, by Monte and colleagues. They take an image, they run it through bandpass filters, think of something like the retina. They also measure local luminance, and they divide here they use a circuit model to describe their process. This is an adjustable resistor, so they're going to adjust the resistance or the conductance of this thing based on this local luminance signal, and there's a capacitor here also, and that's going to create not only an adjustment of the gain, but also it's going to affect the dynamics of the circuit. It's going to make things faster or slower, and those are things that we see in these neurons. And so they've tried to capture a little bit more than just the fact that the gain gets adjusted. They're trying to capture things like phase delays and changes in dynamics. They do another gain control. So this is a luminance gain control. Here they take a pile of neurons that are responding to this luminance normalized signal. They take those, they square them, they average them, and then they take the square root and they call that the local contrast, and they normalize by that in another RC circuit. So there's two RC circuits in serial. I had two RC circuits in my cereal this morning. I think it was a German thing. Anyway, resistors and it's muesli. It's an RC muesli. Nobody's laughing. I'm making nerd jokes and nobody's laughing. This is a nerd course. If you can't make a nerd joke in a nerd course, then where can you make a nerd joke? Okay. So then there's some noise, and then they rectify. Of course, they've got a rectified linear unit at the end, and then they're done. Okay. So, and here's actually a model that I did. Actually, this is my thesis project with Ted, and it was part of my thesis. And then David Heeger and I published the sort of physiological part of this a few years later. So this is a stacked version of this. So here's a V1 stage that's doing local normalization. And then there's an MT stage that combines over a bunch of V1 neurons and does yet another round of this kind of gain control and normalization. So that's what we want to, at least that's what I want to go for. I want to think about how we can stack these things. What properties do they have? How can we fit them to physiological data? How can we understand their perceptual consequences? And how do we think about really their computational consequences? So one thing that you got to wonder here is, a lot of you are on the machine vision side, so you're probably thinking, well, is this something I need to care about? In other words, is this something that's there just because it's, made of, it's got channels and ions and membranes, it's mushy stuff. And so it's just there because it's mushy stuff, and this is how you implement things with mushy stuff. If I were implementing it in silicon, I wouldn't bother with all of this. I would just set all the gains correctly, and then I wouldn't have to worry about it. So the question is this gain control stuff I'm talking about, is it computationally essential, or is it just there to maintain a biological system? I think it's computationally essential, and I'm going to try to give you some examples of that. Okay, so first example, and this is going to be a perceptual example. If we want to quantify visibility of image distortions, you heard some of this from Ruth, you heard a little bit of this from Bill, so I'm going to give a different kind of variant on this. A standard choice in image processing and computer vision would be to measure the mean squared error. So here's two images. There's an original image. Here's a really messed up version of that that's been highly compressed. And it looks, and what we would normally do is we would tune our image processing systems or our whatever processing we're doing on a computer to sort of minimize this. We want to make this thing as close to that thing as we can get it. The problem is that mean squared error is not actually a good description of the visibility of errors, and you can convince yourself that really easily. So this is an example where that's an original image in the middle, and here are 8 images. Each has exactly the same mean squared error compared to that original image, and I'm sure that you don't see them all as equally bad. And I'm sure that you see that one as really terrible, and you probably see this one as pretty bad, and you see that one as pretty bad, and this one, I don't know, this one looks almost as good as the original. Maybe you lost a little bit of stuff in the shadows, but that's because this room is too bright. This one's a little bit maybe reduced in contrast, but not too bad, etc. So the point is, these look very different to a human, but to a mean squared error viewer or subject, they look the same. So we need a better metric. And why does mean squared error fail? Well, it's sort of obvious. The human visual system is computing things. We're not computing something linear. If we were, then a variant on mean squared error, like a weighted mean squared error, would do the job. Maybe it would be like a frequency-weighted mean squared error. And in fact, if you do that, it is better. It's better than plain mean squared error, but it's still not that good. So the point is, you're doing something in your brain that's very nonlinear, and roughly the idea is that when you detect errors in these things, you don't detect them with your retina or with your cones. You detect them somewhere very deep in your brain after all that nonlinear processing. So that suggests that what we should do is replace mean squared error with, I don't know, let's keep it simple, mean squared error in some other domain. And the other domain is going to be something that we get to by doing some processing on the image, nonlinear processing, and the nonlinear processing probably should look something like what the visual system does. So let's try it. So there's a static gain control model of the LGN. This is kind of like that Monte model, but it's been simplified. We got rid of the RC circuits, and all we're going to do is, okay, what are all these boxes? That's a center surround filter. This is a low-pass filter that we're going to compute the luminance with, and we're going to divide by it. That was also in the Monte model. Then we're going to do another division. We're going to square the responses of this thing, again, blur them, and now square root them and divide by that, and then we're going to rectify the whole thing, and that's the output of this channel. This is an on channel, on center channel. Here's an off channel. It looks exactly the same, except that it's got an off-centered receptive field. Okay, so this is an on and off model of the LGN. It captures the basic response properties and gain control kinds of things that you see in the LGN, and it has, for our purposes, we're going to think of it as a parametric model that has a handful of parameters. I think it's a dozen parameters, like 2 parameters to describe the size of the center and surround, a parameter for the size of this, a parameter for the size of this, a couple of parameters for the nonlinearity, separate parameters for the off stage, and you put that all together, and I think it's 12. 12. It's 12. Oh, it's on the next slide. Good. There it is. 12 parameters. Okay. Now, what we're going to do is we want to compare this. We're going to ask about the perceptual consequences, and when we do that, we're going to ask in comparison to something else. So, since we live in the world of deep nets, we decided to cave in and compare to a deep net. 2 deep nets, actually. So here's VGG trained on recognition. And we're going to take mean squared error, but we're going to do mean squared error at 13 different stages of the deep net. And then we're going to take a weighted sum of those 13 mean squared errors. We're going to take all those squared errors, average them together with some weighting, and that'll be our measure of error. And the parameters of that model are going to be the 13 weights that we're going to use to average together the mean squared error at each of those stages. Okay, so 13 parameters, 12 parameters, and then we'll also train a generic CNN. I can't remember how many stages, but it has 500,000 parameters, which used to make me wince, but now I just say it like, it's not, it's a baby one. Okay. Now I just smile. Okay, so we take these models, and all three of them are going to optimize the same way. We're going to take a database of human ratings of distortion. How bad is this distortion on a scale from 1 to 10? And this is a database with many observers. I can't remember the number of people. And lots of different distorted images. So they get to see an original and a distorted image. How bad is the distortion? And they have to pick a rating. And we're going to fit each of these models to those data, okay? And each of them, as it turns out, describes the data really well. So this is the correlation of each of these three models. They're color-coded with the data in that database. It's cross-validated. And you can see that the performance of the three models, here's mean squared error, the black bar. Here's the three models. They're almost the same, but they do come out in the order that they're shown here. That is, the on-off LGN model is the worst by a little bit. This VGG model is a little better, and the CNN model is a little better. You better hope so with 500,000 parameters. So it's a little bit better, not much. So you could say, okay, victory for the CNN. We're done. But these are describing the training data. The training data only contains a limited set of images with a limited set of distortions. And we wanted to know, what do they predict beyond that training data? And in particular, we wanted to really push them to predict something that's way beyond the training data. So we didn't want to go out and gather some more distorted images and say, gee, can we get the right answer in these? What we did instead was sort of more of a theorist's version of an experiment. So we're going to generate images from each of the models. So this is a trick that we've been doing for ages in my lab. It's kind of, if you like, it's sort of the theme of what we do. We're going to start, here's an image. Imagine that the nonlinear transform that one of these models is doing puts us into some response space. R, it's a vector space. So here's these two axes are pixels, pixel intensities, and these two axes are response intensities in whatever the space is for the model. And we're saying we're going to use mean squared error in model space. So that means that all the points on a circle like that around this central response are equally bad, right? And of course, this is 2 dimensions, but the actual model spaces are much higher, and same thing for the pixel space. Everybody knows that. This is a 2D cartoon just to show you what's going on. So because this is a circle, but this transformation is nonlinear, what this circle implies is something more elliptical back here in the original space. And actually, it wouldn't even really be elliptical, because if this is a complicated nonlinearity, this shape could be pretty strange looking. But we're going to do a local approximation. Think Taylor series. You imagine that locally, think of the Jacobian, right, as a local linear approximation of what this transformation is going to do. And if it were linear, it would map a circle back to, or it would map an ellipse to a circle, or a circle back to an ellipse if you went this way. So what we're going to do is we're going to calculate this elliptical shape. What does that mean? Why would we care? Well, this elliptical shape is telling us that not all directions are equally bad. In fact, if I move just a little bit in this direction, the short direction of the ellipse, that puts me on the ellipse, and that error, which is a very tiny error back in the pixel space, is just as bad as moving a huge distance from here out to the edge of this long axis, okay? And in fact, because this is in a very high-dimensional space, you have to imagine a high-dimensional ellipsoid, which has many axes of different lengths. And what we want to really do we're not going to measure all those, because eventually we want to show this to human observers. What we have to do is we have to come up with an economical way to describe the situation. So what we're going to do is take the extrema. We want the longest axis, and we want the shortest axis. Everything else is in between those. So we're going to find the worst case and the best case, and we're going to let the model tell us what the worst case and the best case are. We can't ask the human to tell us that. We can't have a human search through that huge dimensional space, but the model can do that. We can do it using GPUs and propagating gradients and doing calculations and using things like the power method to find the eigenvectors, for those of you that know about the tools here. We're going to look for the eigenvectors of the Fisher information matrix. That's the technical description of what we're going to do. So we calculate we have a representation of the Fisher information matrix, which is basically just that ellipse, and we're going to use the power method to find the largest eigenvector of that ellipse. We're also going to use the power method to find the smallest eigenvector of that ellipse. Those are the shortest and the least and most noticeable discriminations according to that model, and then we're going to show those to human subjects. Okay. So mean squared error, here's a plot of thresholds for human subjects. So now we've gone and done that on the 3 models. When we show the stimuli to human subjects, if we just measure, if we want to know about mean squared error is a circle and it stays a circle. There's no transformation. The transformation is the identity. So all directions are equally bad or equally good. So we're just going to add white noise and measure how easy is it for humans to detect the white noise. And we get an answer for a threshold. How strong do we have to make the white noise for them to see it reliably? And we get this. Here are the least noticeable distortions according to the 3 models that I told you about. You probably can't see anything given the contrast of things in this room, but maybe you can just barely make out that these 2 look kind of like noise, and this one looks not quite like noise. It looks like it's got some little bumpy things, and they seem to be kind of aligned with the lines on the parrot's face. And that's exactly what they are. They're basically masked, so they're not very visible. And when you go and you measure thresholds, you find that out. In order for a human to see this thing, I have to make it really big. I have to amplify it by a lot. That's what this point represents. This is a log plot, so this is at a value of 20. These 2 are back down kind of near the white noise. So if I make those at values, at amplitude values kind of near the white noise, they become visible. Okay. Now these are the most noticeable distortions, again, according to each of these models. And you can see what those distortions look like. The on-off model puts these 2 big spots right on the parrot's neck. I don't know why it puts 2, but it does. The CNN puts these funny high-frequency liney things down on the parrot's neck. Everybody goes for the parrot's neck. It's like go for the jugular. And the VGG network puts some sort of funny, noisy thing down on the parrot's neck again. I think the parrot is most affected by this one. And humans certainly are most able to notice this one, and that's why this threshold is down lowest. So you have to, in order for you not to be able to notice that, I have to make it really, really dim. And that's what this number means for our 19 subjects, and these 2 not quite as dim. So the critical point here is that remember, these are the minimum and maximum. Each model is being asked, what is the most visible thing you can add to this image, and what is the least visible thing you can add to this image? So what we're really looking for is for the 2 numbers to be as far apart as possible. And what you can see here is we're like orders of magnitude off here, right? So this is 20, and this is 0.05. Okay, so it's a factor of like 400 of intensity from the minimal one to the maximal one in the on-off model, the LGN model. And the deep nets are doing something, but they're not nearly as much in terms of that range. Okay? You might ask yourself, well, did you really need that whole model to do this? And we can sort of break it down to simpler models. This is the full model. If we get rid of the on and off stages, we just have an on stage, then we get these 2 values. If we get rid of one of the gain controls, then we get these 2 values. So they're getting a little bit weaker in terms of their spread. But if we get rid of the gain control, we go from this LG down to this LN. Now it's much weaker. Okay? And if we get rid of everything, you're back to the mean squared error. That's this point. Okay? And you can see that the deep nets, and by the way, we've tried this on other deep nets, and they all have similar properties. You can see that the deep nets are they're a little bit better than the LN model. I would hope so, because the LN model is a shallow deep net. It's a 1-stage deep net. So these are a little better than that, but not a lot. Okay? And the 3 things that have gain control are all working quite well. Okay, Ruth.
Can you just clarify, you showed an example image on the last slide, and was the data for that one image or for some number?
No, sorry, the data were averaged over all the images that was just one example, so you could get the picture of what.
How many images? That's what I'm asking.
I think it was on the order of 20 images. So it's not a huge number. We're a little lame when we do our psychophysics. You already know that. I'm confessing many things here today. I hope you're all taking note. I will go out of here and say a lot of Hail Marys or whatever I'm supposed to do. I told you I was Catholic when I grew up. Go ahead.
So, Eero, when you say the LN model is a shallow deep net, that means you're training the filter and the weights.
Yes. Yes.
In the model you just showed us, you are not, you have these 12 parameters only, you're not training the full filter and all that.
That's true, that's fair. The filters were restricted.
You could
You're right.
more parameters in the same architecture.
Oh, yeah, so maybe you're right. Maybe the Allen model would beat the deep net if we allowed it to be an arbitrary filter. That's good observation. Okay, let me do another one. We're going to run out of time. Got to move quickly. I'm going to take that same idea, I'm going to use it for a practical problem, which is image rendering. So here's the image rendering problem. I already mentioned it with the video camera example. The range of intensities in the outside world is this blue bar. It's huge, right? From dark shadows, if I put my head down in this trash can, which I think I'll do, and speak to you like this. No. It's really dark, and if I look at that, it's very bright, and there's a huge range, right? So what happens? Well, your visual system actually is quite good at perceiving most of that range, although it often needs some time to adapt. And it adapts actually over many time scales. There are things you can stand in this room and see the stuff that's down in these shadows and the stuff that's coming from that window at the same time. But if you take out your cell phone or your camera and you try to take a picture that includes both of those things, it's not going to work out too well. And I think you probably all know that from experience. So why is that? Well, the camera kind of has very limited dynamic range, and it tries to adjust shutter and aperture speed and ISO in order to get the dynamic range good enough to handle all those things, and it never really works. When you go into high dynamic range mode on a camera, what they usually do is they take multiple exposures in rapid succession with different sensitivities, and then they blend them. But the problem still remains. Once you've blended them, now you have this high dynamic representation, but you have to put it on a screen. And the screen is low dynamic range again. So how do you put something on this screen, or that screen, have it look like what's in the outside world, knowing that the screen has this little tiny range? So here's a typical screen range. It's a couple hundred candelas per meter squared. It'll get that bright. And here's the stuff that you're trying to actually render or display. How do you do it? Well, there's kind of dumb answers. You can take the giant thing from the outside world and just squeeze it down to fit on the screen, and that doesn't look very good because you really won't see anything in the shadows. You can run it through some nonlinear operation, mapping the intensities into the pixel intensities, and that's a very common and standard thing. It's very easy to do. You can do it in Photoshop. You play around with curves and things, and that gets you somewhere, but it doesn't work that well. So the question is, what can we do better? Okay, and whoops. Oh yeah, when I was giving this talk a couple of years ago, or a description of this maybe about 2 years ago, I was standing on the balcony that morning before I went to give my talk, and I realized it was a perfect example. So this was a bright but hazy day in Mexico, and I took a picture with my camera, and I couldn't see anything here, even though I was standing on the balcony. I can perfectly well see, the rendering here is even worse, but I could see bugs on the leaves in here. I could see all the details in the shadows. I could see everything in this tree, in this bush. It was right in front of me. And at the same time, I could see all that stuff in the distance, but my camera couldn't see any of this. So I went in, this was like adjusted in Photoshop and was as good as I could get it. It looks better on my screen, but it still looks pretty bad. Okay, so that's the problem. So this is an engineering problem, and what we're going to try to do is just use the thing I already told you about. It's slightly different, but I'm going to gloss over the details. We're going to take, what we want is not to make the thing on the screen equal to, or which is impossible, to the real world, nor to make it minimize the mean squared error with the real world, because what that would actually give you is just to truncate all the high values. If you think about it for a second, what's the best mean squared error solution to this? It's a ridiculous solution. It looks terrible. So what do we want? We want to minimize some internal representation. I want my internal representation of the real world to be similar to my internal representation of the thing that's going to go on this screen. So I'm going to have to solve an optimization problem. I'm going to have to search for an image to put on the screen that satisfies two things. First, it minimizes this distance. And second, it has to be something that can live on the screen, right? It's a constrained optimization problem. I can't just do anything. I have to make it live on this screen, whatever the intensity ranges are and everything else. Color limitations, whatever the limitations are, I have to live with those. Okay, so that's the problem that we're going to try to solve. And this is not a real world, like a real engineering solution, because it's slow. It's going to require a massive optimization over the space of images. We're going to optimize over I, the image, but it works really well. So here's an example. I'm just going to show you a couple of examples. So that's an original high dynamic range scene. It's Yosemite. The falls are very bright because the sun is on them, so they're basically white. If I just linearly rescale that into the range of intensities, and I should have calibrated my screen a little bit better because I don't think these are going to come out as well as they should. But anyway, if I just linearly rescale, I get that. You can see the waterfall, and you can't see much else. It's not quite as bad as it looks on this projector, but it's still pretty bad. This is a state-of-the-art algorithm 2015 in SIGGRAPH that describes a sort of heuristic procedure that's very clever for readjusting things locally to have them look better. And this is what comes out of our optimization. This is completely automatic, so we didn't set any parameters. We took this model F, which is our description of the gain-controlled representation, and we locked it. We didn't touch it. What we did then is ask the computer, please optimize this image for display on this screen, except I didn't do it for this screen or this projector. I did it for a generic display that has a range from 0 to 250 candelas per meter squared. Okay, so that's not this display, but it's close enough. And it works really well. You can basically see detail throughout, and yet you still see that the waterfalls are much brighter than the other stuff. And if you want to come up and look at it on my screen later, you'll get a better idea of what it does. Okay. You can use this for dithering. So if you have, let's say that your display is it's paper, and you can only put black ink or no ink. Those are your options, black or white. You don't get grayscale from some range. You get black or white. How do I arrange the black and white pixels so that it looks good? There are many standard algorithms in the literature. The most standard one is the Floyd-Steinberg method. If you use our method to rearrange those black and white pixels, and I'm glossing over tons of details here, but you can get something that looks much higher contrast and captures a lot more of the detail and structure of the original image. And you can also just use this to enhance contrast. So if you pretend that you have a screen with smaller range and ask it to display on that, it's funny, because the luminance range is smaller, what it does is it boosts the contrast to make the image look more, I don't know, salient to your visual system or to your LGN. And so here's the original, which looks reasonable. Here's one that's been boosted a little bit by telling it that the maximum intensity on the screen is 1,000 candelas per meter squared. The maximum intensity in the scene is 1,000 candelas per meter squared. Here we make it 10,000, and here we make it 100,000. And as this thinks that it's taking a brighter and brighter image and packing it onto a smaller screen, it makes up for that reduction in luminance range by boosting contrast. And so this sort of happens pretty naturally and pretty automatically. Okay. I'm running out of time. Yeah.
Can I ask a question? Since the results are beautiful, but you said it's not practical and it's slow down.
Not in its current form, yeah.
So what I would do is I would take your method, I would generate lots of training examples, input and output, and I'd train a neural network to solve this problem.
Code's online. Go for it.
What does that neural network look like in the end?
I have no idea.
Would it look like the thing you started with, right? And
No.
so is that interesting or?
No, okay. If you just want to solve the engineering problem and you want to assume universal function approximation, you say there's some function that maps this to that. All I need to do is to implement that function. I'm going to use a deep neural network with 1 billion parameters, and it'll figure it out. Because I can generate as much training data as I want. Sure, go for it. I'm fine with that. We don't have to understand it. We don't have to know what function it implements. Well, we do know it implements this function. But the way you got this function was by modeling the human visual system. So yes, you could do that. That would be great. Good solution. It might also be, though, that if you looked inside that box, you would find out that it's doing maybe something like an autoencoder that has to do some sort of gain control and then invert it back in some way that knows about squeezing it into the output range. So that would be, I think, more interesting. But the solution would be the same. Yeah.
I maybe missed the details, but is this the same form of model as you showed earlier?
Yeah, you missed the details because I intentionally hid them from you. And now you have unmasked me. Yeah, fine. It's using the same kind of gain control model, but it's multi-scale. I slipped that by and didn't say anything about it. So it's inside of a Laplacian pyramid. And then there are some other details that are probably not worth mentioning at the moment. Yeah.
Following on Michael's question, if you did that with property, might it be brittle?
I mean, my old school answer to that is it might be brittle, but that's mostly because it's basically under constrained, and you can fix that by just generating more training data. And here, since you can generate an unlimited amount, hopefully, eventually, you would solve that problem. You know, I don't know that I completely believe that. There's this thing called the Curse of Dimensionality. You're in a big space. You have 1,000 by 1,000 pixels. It's a million-dimensional space. In my old self, 5 years ago, I would have said, there's nothing you can do. You will not get enough data to fit a complicated model in a million-dimensional space. You're just never going to get enough data. Doesn't matter if you can generate as many examples as you want. You just can't fill up that space. It's too big. And I guess I don't know if I believe that anymore, because the whole world seems to have just discarded it, and I'm now in a state of confusion. If anybody can help me, I'm please. I'm confused. All right. One more example, and then I think I'm going to be out of time. I had 2 more examples, but I'm going to be out of time. Okay, so now we're going to build another application, and this one is going to use, this one will be a little bit closer maybe to what Michael was asking for. We're going to build an application, we're going to put gain control directly into the application. It's not because we're mimicking biological vision in any explicit way. We just want to know, if I put gain control into one of these networks, what happens? So I want to do compression. Compression is a very old problem. It's an extremely well understood problem. The basics go back to Shannon in the 1940s. So we have deep roots here. We understand the problem well. And I want to ask, can we tackle that problem using a combination of sort of deep net optimization technology and gain control from biology? So how does a transform coder work? Basically, the idea is you take your input, you transform it into some other space. It could be a Fourier transform or a block DCT if you're talking about JPEG, or it could be a wavelet transform that uses oriented filters. Whatever it is, you transform into that space. You quantize things in that space, i.e. you convert them from continuous values to discrete values using some sort of step function transformation. Why do you need discrete values? Because you're going to code them with an entropy coder that can code a discrete distribution. That's what Shannon told us how to do. He explained all of this. He defined entropy. He described what it means to do compression. So we're going to measure the entropy of this compressed format after we're going to measure the entropy of this quantized format, and then that's the thing that will get bit coded and sent as a code to represent the input. On the other end, the decoder will have to take those quantized values and run them back through maybe the same kind of transform, invert it, and reconstruct an image. Okay, so what happens when you do coding? I hope you can see these well enough to see all the artifacts. So these are JPEG and JPEG 2000 coded images, and they're coded at a very low bit rate. When you lower the bit rate, the quantization levels become very coarse, and you can see the errors. In this JPEG one, you can see blocking artifacts. You see these little square things. This is a nice smooth car, photograph of a car in Cuba. You can tell it's Cuba because it's a car from the 1950s. It's not quite as old as Shannon, but it's close. And the blocking artifacts are just horrendous. If you saw it on my screen, you would see that it's much worse than it looks like on this projector. And down here are JPEG 2000 artifacts. JPEG uses block DCT for its transform. JPEG 2000 uses an orthogonal wavelet. Don't worry if those words don't mean anything to you. Just think of this as carving things into blocks and then transforming them. And this doesn't carve them into blocks. It's more convolutional. Okay, so what you see in these coders are artifacts that look like the basis functions. The blocks are showing up here because each block is being coded independently by these blocky basis functions, and when you throw away a lot of information because you've quantized it and you've thrown away the high frequencies and things like that, the blocks no longer match up correctly at their boundaries, and you see the boundaries of the blocks. Here you don't see boundaries of blocks, you see basis functions that are sort of popping out. You can see one right there. It's just like it jumps out like a bug on the wall. Because that one was a little bit over threshold for the quantizer, and so it ended up surviving, but all the ones around it got thrown away. So there's not some sort of gentle way of controlling these things together to have them fade out or come in. You just decide on each one, I keep it or I get rid of it. And the quantizer is making these kind of abrupt and very discontinuous decisions about whether to keep things or throw them away. Okay, and that's a hint as to why gain control is going to help us, because what we're going to do is we're going to take representations like this of local features and we're going to gain control them together so that they're going to go up and down together, and if they get quantized in or out, they will all get quantized as a group together or get thrown away together. We're going to not do that explicitly, we're going to do it deep net style. We're going to build here, I'm going to build a representation like this. So here's the image, X, it goes through some transformation to produce Y. And Y gets quantized. We measure the entropy or the rate of the coder at this quantization stage. Then the decoder has to take this Q, it has to reconstruct a continuous valued Y, it has to run it through an inverse transform, and that gives us our reconstructed signal X hat. And then we compare X and X hat and measure the distortion. So we have two things that we care about here, the rate, how much is it going to cost me to send this image, and the distortion, which is how good is it going to look or how bad is it going to look, and those trade off. Okay, so there's a rate distortion trade-off. This is completely classic, again, goes back to Shannon. These two things trade off. There's some curve that describes the trade-off between rate and distortion. If I increase the rate, I can reduce the distortion. If I decrease the rate, then I'm going to increase the distortion. There's no free lunch here. You have to make a decision about where you want to be along that curve. We're going to try to solve, so the optimization problem becomes, well, there's two terms in the objective function. There's a rate term, there's a distortion term. You've got a lambda in front of it. The lambda is basically describing the slope of the line, the tangent line of the point that you're aiming for, okay? This is all basic, the geometry of that trade-off. By the way, you see these same kind of trade-offs throughout economics. Utility theory goes back to the early 1900s, before Shannon, and describes this kind of trade-off and the sort of optimality principles, Pareto optimality principles in the early 1900s, the idea of how do you allocate goods to people given the value of the good and the costs. Okay, so optimal allocation of bits and the optimal allocation in order to minimize distortion, that's what this game is about. So we're going to train a deep net to do this. The deep net is going to have 3 stages, not 20, not 50, not 100, 3 stages of architecture, 1, 2, 3. The difference from a standard deep net is that we're going to do divisive normalization. So we're going to have local normalization, we're going to compute, we have convolutional filters, there are 128 of them. We're going to, at each stage, divide these guys by their local neighbors, a weighted sum of their local neighbors, that's what this thing is down here. And that weighting, we're going to learn not only the filters, but also this weighting. And we're going to do that in 3 stages, 1, 2, 3. There's about 1 million parameters, which again used to make me shudder, and now I just say it and move on. So there's the forward transform, and here's the inverse transform. It does something kind of like the opposite of that, but not exactly. It's just, it multiplies things back out. Okay, so we have these, yeah.
So nearest neighbors means within channel.
Nearest neighbors means here, both within channel and across channels, and it's spatially localized. Okay, so it is across channels. It's across, I'm pretty sure it's across all 128 channels.
I see.
Yes. As it would be if you were thinking of visual cortex. So imagine that this thing has oriented filters in it. Each oriented filter is divided by not only its neighbors of the same orientation, but also by some other orientations and some other scales.
Right, but you're not taking into account similarity between your...
No, we're going to let it learn that. We could build that in, and that would save us a lot of parameters, but we're not going to. We're going to let it learn it. It's going to have to learn those weights. So we're going to have to show this a lot of images. Fortunately for this problem, all you need is a nice, plentiful supply of clean images. That's got a nice feel, huh? It's like all the world needs is a plentiful supply of clean water. So I need clean images, that's all. And then I can run them through this, and I can measure the rate and the distortion, and I'm going to optimize that, okay? There's one more piece. It's a little bit technical, but I'll just say it for some of you who might be interested. There's a quantizer in this system. A quantizer is really bad for optimization because if I want to do gradient descent, the gradients of that thing are, the derivatives of that thing are either 0 or infinity. Those are the 2 bad cases, right? 0 means I'm not going to go anywhere, and infinity means I don't know which way to go. So I don't want 0 and infinity. I want nice intermediate values of gradient. And so what I'm going to have to do is replace that quantizer for purposes of training and optimization with something else. So what we did is stochastically, replace the quantizer with additive uniform noise. It's not the same, but it's quite similar in its properties because the errors that you incur in a quantizer are deterministic, but they're always kind of within some range, right? I either am in this bin or I'm in the next bin, so the bin size decides how big the errors are. If I add uniform noise, it has a similar kind of error behavior, and in fact, there's a long history of people doing that to model errors in coders, not for optimization purposes, which is the way we used it. So we're going to use stochastic gradient descent, so it fits perfectly because we're going to add noise instead of quantizing, and we're going to optimize the entire system for that with the additive noise. It turns out that when you go through this and write it all out, it's a variational autoencoder. Except this variational autoencoder is one that goes back to Shannon, so it's not one that was invented last year. It goes back to Shannon. It is a rate distortion optimization procedure, and the only thing that's different from classic rate distortion is we've substituted this noise for the quantizer. Okay, so, and if you want to think about it as a variational autoencoder, this rate term is a log prior, and the distortion term is a log likelihood. Okay, I realize that for half of you that means nothing, but that's okay. This slide was the only technical slide. All right, so we go ahead and we do it, and it works really well. It's state of the art. It was when we did this. I told my postdoc, Johannes Bala, said he wanted to do this. I told him he was crazy. I said it probably won't work. He said we have to try. I said please don't do this. I hate deep nets. He said we have to try. It'll be interesting. I said okay. I give up. And I was completely amazed. It was stunning. So I told you about transform coders and how you can see all the artifacts, the blocks, the wavelets. Not in this. So on the left, these are 3 images coded at the same rate by JPEG 2000 on the bottom, and our coder in the middle. And over here, they all look pretty good. Over here, they start, I don't know if you can see it, but the top and the bottom ones start to show some weird artifacts. There's some blocky things showing up here and here. And by the time you get over here, I hope everybody in the room can see this looks ridiculous. This also looks really bad, although maybe not quite so bad to where you're sitting. And this one looks almost as good as the original, but what's strange is it's like gives up on representing some features, but when it does it, it completely gives up. It doesn't keep like half a feature there. So look at like this little yellow thing right here. Okay, so this is one of these little, what do you call those? In a flower? The thing at the end.
Filament and stain.
Not too many biologists in the audience.
Anther.
I'm going to check that, but anther. And then the anther. Note the anther over here, which has vanished over here. It's just removed. Note that this leaf has a little kink in it up here. Over here, let's just smooth it out. We'll just close the contour. We can't afford that little detail in the contour. We'll give it up, but we're not going to blur it out or turn it into some blocky mess or add some weird little wavelety artifact. We're just going to smooth it out. It's pretty amazing that the distortions. This, by the way, was trained for mean squared error. It was not trained for some. This is one of the mysteries. We did not train this for visual perception, and yet it looks fantastic. Here's another one, the car I already showed you. That's the original JPEG 2000, and this thing, and this is, again, you can't see it too well on the projector, but there are a lot of artifacts, especially around curved contours in the JPEG one and in the JPEG 2001. I don't know if you can see those, but along the curved parts of the roof, and they're just completely gone when you get to this. And again, it eliminates some features, but the things that it keeps, it does well. It's even more amazing in this one. So this motorcycle, I hope you can all see, there's sort of a plastic cowling over the shock absorbers here that's got little stripes in it. JPEG sort of tries to get that and puts it in aggressively in a few spots and then mangles it in other places. JPEG 2000, same thing. This thing just decides, you know what, you haven't given me enough bits to represent that. So it's gone. I'm going to get rid of it. And it just smooths it out. It makes a nice smooth cowling, as if your motorcycle came from a different manufacturer. But otherwise, everything is fine. So I think actually it's well, anyway, I said enough. I think it turned out pretty amazing, much more than I thought. You can quantify the performance, plotting PSNR as a function of bit rate. So better is up in this corner, right? That would be lower bit rate, and higher PSNR. This is signal to noise ratio, so big is good. This curve is over, I think it's over the whole Kodak image set, which is, I don't know, 20-something high-quality images. And these are JPEG and JPEG 2000. And this is not quite state-of-the-art now, although I think visually it's still state-of-the-art. In terms of PSNR, it's a little bit worse than, there's some new networks that are doing a little bit better than this, but I think visually this is still competitive. And here it is in terms of multi-scale SSIM, which is a metric we made many years ago for visual quality, just showing that if you try to measure it with something not mean squared error, it also greatly outperforms the other two. Okay. I think my time is up, so I'm going to skip this last chunk and jump to my conclusion slides. And there's only like another 50 slides to go. That wasn't so bad. I'm joking. It was for EJ's sake. Okay, so I tried to convince you that simple canonical neural models that include local gain control can predict human perception of visual distortions, can account for perceptual straightening of natural videos. I didn't show you that. That was the part I just skipped. Can be used to optimize image rendering systems, and can be used cascaded for image compression. And I think this is really just the beginning. I think we're just starting. We still don't really understand the capabilities of these kinds of computations. We don't really understand what happens when you stack them. We don't really understand how they show up in the entire biological chain. We've documented the existence of these things in certain patches and spots, but it's not really been done sort of fully and systematically the way it should be. But I think that's the way science proceeds, that's the way engineering proceeds. It's kind of successive approximations. All models are wrong, and we're going to keep evolving things and trying to keep them simple. In my view, I hope that we can keep them simple and understandable. I find myself completely overwhelmed by the deep net era because the canonical elements are simple, but the results are completely not understandable. So I'm hoping that you young folks are going to figure that out and maybe explain it to me if there's still time. Okay, thanks.